Hello everyone, thank you for making an effort to watch our video. We are students from Sunway University which are going to present about the interaction between the microorganism and the environmental object. Before going through this presentation, let me first introduce my group member. I'm Joey and my group members are Dineka, Dennis, Jensen and Chenya. We are currently taking the same course and the course instructor is Dr. Huan Jun San. The topic that we are going to present today is Methanococcus Jannaschii and hydrothermal vent. Our presentation is divided into 4 parts, which is introduction, description of Methanococcus Jannaschii, discussion, and conclusion. So let me start with a brief introduction about the title and the rationale of selecting the hydrothermal vent as our environmental object. The environmental object that we choose is hydrothermal vent. Hydrothermal vent is the eruption of water heated by geothermal and it's cracked from the seabed. It is divided into 2 types, which is black smokers and white smokers. It also usually found in deep oceans with frequent volcanic activities and shifting continental plates, as well as near sea basins and hotspots. And hydrothermal vent has unique characteristics. For example, hydrothermal vent has extreme environmental conditions of high temperature, high hydrogen sulfide, low oxygen content, and low pH. Hydrothermal vents were surrounded by large number of organisms that managed to adapt the extreme environment. Methanococcus jannaschii was initially discovered in the East Pacific rise near Mexico western coast in 1982. The bacterium was discovered in a sediment sample taken from a 2,600-meter deep white smoker chimney. It lived in a severe environment with temperature ranging from 48 degrees Celsius to 94 degrees Celsius and a pressure of 200 standard atmosphere and a pH of 4.2 to 7. So, the rationale we choose this environmental subject is because we want to know about how microorganisms adapt themselves in this harsh environment and the role they play in the hydrothermal vent ecosystem. Besides that, we are also less likely exposed to underwater volcanoes. Thus, we wish to understand the relevant knowledge about the microorganisms which inhabit nearby the hydrothermal vent. Now, I'm going to hand you over to Jensen, who is going to talk about the description of Methanococcus jannaschii.
Hi, I'm Jensen, and I'll be talking about the description of Methanococcus jannaschii. Methanococcus jannaschii is a member of the genus Methanococcus. It's from the domain Archaea, and it's from the kingdom Euarchaeota. M. jannaschii is an autotrophic single-cell organism, which means it's an organism that can produce its own food using light, water, carbon dioxide, or other chemicals. It has an irregular cocci shape or irregular spherical shape, and unlike most bacteria, the cell walls of the M. jannaschii is made of proteins or glycoproteins instead of peptidoglycan. As for the cytoplasmic membrane, for other species, the membrane is made of phospholipids, but for M. jannaschii, the cytoplasmic membrane is made of glycerol. It has a sequenced genome and proton. As for proton, it means that it's an expression of an organism's genome. And lastly, it has polar bundles of flagella, as you can see on the slide, to move around. As for the genome structure of the M. jannaschii, it has 3 characteristic elements, which is the large circular chromosome, the large circular extra chromosome, and the small circular extra chromosome. For the large circular chromosome, it has a length of 1.66 megabase pair, and within the chromosome, there are a total of 1,729 protein coding regions, and the percentage of GC content is 31.4%. As you can see on the right, the GC content, also known as guanine cytosine content, is the percentage of nitrogenous bases in a DNA or RNA molecule. They are either guanine or cytosine. So moving on, the second characteristic elements that make up the genome is the large circular extra chromosome. It has a length of 58 kilobase pairs, and the total protein coding regions are 45. And for the GC content, it's reported to be 28.2%. Lastly, we have the smaller circular extra chromosome. The small circular extra chromosome contains 16.5 kilobase pair, and the predicted protein coding region are 12, and for GC content, it is 28.9%.
And now I'm going to talk about the growth conditions of Methanococcus jannaschii. First, this bacteria is hyperthermophilic, which means the organism can thrive in very hot environments. And the optimum growth temperatures for Methanococcus jannaschii is 85 degrees Celsius. In this graph, for the blue line is the optimum growth temperature for hyperthermophilic. And secondly, the bacteria is also barophilic, which means the organism can survive in a very high pressure environment. In this graph, the green line indicates the optimum pressure for the barophilics, which is about an estimate of 500 standard atmosphere, while for the Methanococcus jannaschii, its optimum growth pressure is about 200 standard atmosphere. So Methanococcus, as it can be isolated from the surface material collected from the base of a white smoker submarine hydrothermal vent. Next is the Methanococcus jannaschii role on Earth. Methanojannaschii belongs to a specific group, which is methanogens, and methanogens play an important role on Earth as it is very important to an aerobic environment, as it can effectively remove the semi-final products of decay, such as the hydrogen and fermentation products into methane. So it can be used to reduce the wastewater and the pollutions of water systems across the world. Let's come to the discussion part, which discuss about the relationship between the microbes and the environmental object in depth. Next one is the Methanococcus jannaschii role associated with the environmental object. It can be used for energy production. As it is an autotrophic organism, it is strictly anaerobic and can only use carbon dioxide as its sole carbon source. Hydrogen is also used as an energy source in this mechanism to reduce carbon dioxide to methane. This is the equation. And when methane is formed, it can be burned to use as an energy. More than that, the Methanococcus jannaschii can also balance the ecosystem of the hydrothermal vent by converting the organic compounds into methane to have an arise of the aerobic environment. Moreover, it can help the compounds that exist in the anaerobic atmosphere to escape into the atmosphere by providing them a pathway. Thus, the other organisms can live and thrive in their optimum environment. Now I'm going to hand it over to Jensen.
So I'll be explaining the mechanism that takes place in Methanococcus jannaschii, which is methanogenesis. Methanogenesis is the main biochemical pathway for M. jannaschii to obtain energy. This pathway depends on a rich supply of hydrogen to reduce carbon dioxide to methane. There are 7 steps in total for this pathway, and each step is catalyzed by a specific enzyme which is synthesized by hyperthermophiles, also known as hyperthermophilic enzymes. One interesting fact about this enzyme is that they are optimally active at high temperatures. So for this pathway, first and foremost, carbon dioxide is introduced into the pathway and is ligated to MF, which is an enzyme called methanolfuran. And this coenzyme is strictly involved in methanogenesis only. The methanolfuran lowers the oxidation state of the carbon dioxide, consequently forming formylmethanolfuran. Next, the formyltransferase catalyzes the reduction of formylmethanolfuran to formyl tetrahydromethanopterin by transferring the formyl group on the formylmethanolfuran onto tetrahydromethanopterin. In the third step, the formyl tetrahydromethanopterin is reduced to methanyl tetrahydromethanopterin by the removal of a water molecule. The enzyme involved in this step is tetrahydromethanopterin cyclohydrolase. Next, this pathway involves 2 functional distinct enzymes that are crucial in the energy-producing metabolism of methanogens. The electrons from the reduced coenzyme F420 are then used to produce methylene tetrahydromethanopterin. As for the 5th step, the methylene tetrahydromethanopterin reductase catalyzes the reduction of methylene tetrahydromethanopterin into a methyl group, which will be methyl tetrahydromethanopterin. The S-methyl transferase transfers the methyl group from tetrahydromethanoctyrine onto a coenzyme called coenzyme M. When the S-methyl transferase transfers a methyl group to coenzyme M, it also creates a proton gradient across the membrane by acting as a primary sodium pump. As for the final step, methane is finally produced with the help of methyl coenzyme M reductase.
Hi, I'm Chenya. Next up, we are going to talk about the outcome of interaction between Methanococcus and other organisms. Before the whole methanogenesis process, some fermentation bacteria like Archaea bacteria undergoes fermentation in the anaerobic environment where there is so little oxygen within the hydrothermal vents deep under the sea. They produce organic compounds, namely carbon dioxide, methanoate, as known as the ester of formic acid, and the acetate. And these organic compounds will be used by the Methanococcus who live around the hydrothermal vent. As Jensen mentioned just now, after methanogenesis, the final product will be methane gas, which is both good and bad for nature. In the bright side, a small portion of methane is used by anaerobic methane oxidizing organism. However, in the dark side, a big portion of methane gas escape into the atmosphere as a very potent greenhouse gas. As you can see in this picture, as a summary of what I said just now, the process of methanogenesis undergoes by biological sources like microbes from the soil, and Methanococcus from the hydrothermal vent will produce methane gas. Some of the methane gas will help in microbial consumption, and some of it will escape to the atmosphere as a chemical degradation. That's all from me. Now I'm going to hand you over to Danica, who is going to talk about the conclusion.
I'm Danica, and I will be talking on the conclusion where I'll explain about the impact of the changes on the human and the planet done by Methanococcus. Find out about the impact of changes Methanococcus has done on the planet. So as we already know that Methanococcus Jannaschii does methanogenesis, which is the production of methane. And methane is considered as a valuable energy source, but even though it is considered to be as one, it has arisen the concerns regarding the atmosphere. And as methane is a greenhouse gas, it contributes to global warming, which has been increasing in the past few years. So with methane being released from the ocean and fuel burned by people, the threat is considered to be 25 times much more greater, and this could last for around 100 years. To how humans have made an impact on this. So as we know, humans do produce flatus, which of course contains methane. And a study done recently on human feces proved that it did contain methane. So what was done was, out of 9 samples, 5 contained archaea, which was capable of producing methane. Even results were also found to be positive in the human rectum. So with humans also contributing to the release of methane into the air pollution has been much more severe, and this leads to risk in human respiratory system. We have now arrived to the conclusion of this presentation. So, as it was thought before, life was believed to be non-existent around hydrothermal vents, since sunlight cannot get through into the deep ocean. However, with constant research, it was actually revealed that organisms such as Methanococcus have adapted to these conditions, such as the changing temperature, rapid high pressure, and very toxic environment. So organisms do inhibit the hydrothermal vent through symbiosis from other ecosystems. And although it produces methane, methane still is a greenhouse gas, and this will of course impact the environmental crisis such as global warming. Our references, of course, please do go through them if you'd love to know more. So this is the end of this session. I hope you did learn something useful and enjoyed it. Thank you so much for staying. Please do leave a like.